Good morning, everyone. Thank you, Ludo, for the very warm introduction. I feel no pressure at all. That's perfect. And I'm very happy to be here. I'm also very happy that you are here, even if it's the very end of the conference. I would like to thank the organizing and program committees of JOBIM, of course. And I will present you the work that we do in the team regarding the exploration of microbial ecosystems and what tools we develop to address both the functional and the taxonomic diversity of these microbial communities. So my talk will present you various works ranging from statistical machine learning to machine reasoning and presenting you along the way some numerical models. I will start with addressing the taxonomic diversity of the microbiome. This is my the only minute I will forget about microbes in my presentation. So let's look at these pictures. And with our human brains, we can very easily classify these pictures, right? And this ability is precisely what we try to reproduce with artificial intelligence, isn't it? So if you look at these figures, these pictures, you have forests on one side, on the left and here, and you have grasslands. These are the ecosystem types that you can assign to the data, to the figures. And you recognize those because you recognize some species, some inhabitants of these ecosystems. So here, the trees for the forest, for instance, could also have been the animals that live into these ecosystems. So ecotypes, they harbor specific species. And ecotypes of the gut microbiome, they are called enterotypes. They were introduced a bit less than 15 years ago. They were identified from microbial composition of many cohorts from mostly industrialized countries. And by clustering the microbial composition, researchers had 3 clusters in which individuals fall. One cluster dominated by Bacteroides, which is a genus of the gut microbiome. One cluster dominated by Prevotella, another genus, and one cluster dominated by Rhuminococcus, or more widely by Firmicutes, a phylum. Is to describe what a normal microbiome is, but also to compare individuals. A bit later, these clusters, they were associated to clinical metadata. And a bit later even, well, there started to be some discussions about the number of clusters that occurred within the microbial composition of the gut microbiome, and whether there were 2 clusters, 3 or 4 clusters, or even if there were clusters at all, actually. Maybe it's just a gradient between Firmicutes here and Bacteroides there. A question that I wanted to address regarding enterotypes is what happens between 2 enterotypes, addressing the continuum between 2 ecotypes, 2 ecosystem types. So here, between grassland and forest, well, you have a lot of things in between. And that is what we want to characterize. So for instance, if I show you a few more pictures, well, here you would have a system with, let's say, 30% forest and 70% grassland, and another with half-half. So if you understood this, you more or less understood what I'm going to present afterwards, because we did the same for the gut microbiome. So just to explain you what the data is, it's very usual in the field. It's basically a matrix describing the microbial composition of the ecosystem. So you have the samples here, and you have features describing the samples. So in our case, the features are taxonomic levels, taxa, very often genus level, but there can be others. And in fact, when we have this kind of matrix, the problem is that we have a lot of features. So it's very complicated for us humans to understand what drives the composition and how to compare individuals. So we have several alternatives there. We can change the taxonomic level, for instance, go up to the phylum level, but then we lose a lot of information, but at least we have less features to compare. An alternative is to calculate enterotypes. So what I showed you earlier. So that is hard clustering. So each of the sample will fall into one cluster. And what we wanted to do, well, is to do something in between. So keep the level of information that we have here and here, so the genus level, but make up a model that is more quantitative and describes the samples as an assembly of microbial guilds, of co-occurring features of co-occurring genera. So it's a simplified view of the gut microbiome that we calculate with a machine learning algorithm from linear algebra, that is non-negative matrix factorization. We applied it to a set of cohorts ranging to 5,000 metagenomes. What NMF does is the following. It is an optimization problem that aims at decomposing the original abundance matrix into two matrices that, when multiplied together, will approximate the original one. And these two matrices, they introduce the profiles, the aggregations of features that I mentioned. Here we call them entero signature. So the first matrix here, W, would describe the composition of the entero signatures. And the second one will give you the information about the relative abundance figures like this, showing you the differences between individuals in terms of. So here, signatures that we identified in our large data sets. So if we look at them, you have several peaks here, showing you the aggregations of taxa that occur within the signatures, so co-occurring taxa. For instance, here you have an entero signature that we call ES-Bact, dominated by Bacteroides, but it has other taxa. And if you look at the first 3 months, you have Prevotella, Firmicutes, and Bacteroides. And you might remember that those are the original enterotypes that I mentioned, which makes sense because, I mean, in 10 years, the composition of the microbiome didn't change much. But we have two more entero signatures here, one dominated by Bifidobacteria and the other one by Escherichia. The reason why we have these two additional ones is because our original data set is much wider than the ones on which the enterotypes were cut. We have not also infants, newborn, preterm infants, elders. So the microbiome composition is much more diverse, but we can capture it because we have a more complex model than just enterotyping. So a sample would be a weighted assembly of entero signatures, and an entero signature is a microbial guild. Is it useful at all to perform this model? Well, here you have a PCOA of the 5,000 samples that we worked with, and you have 4 of them, each highlighting the samples that are accurately described by 1, 2, 3, or 4 entero signatures. And you will notice that nearly 80% of the samples, they are best described by a combination of 2 or 3 entero signatures. So a single hard cluster is not enough. We need a more complex model. So since the model is a bit more complex, obviously we capture more variance when we compare to what we have with the original abundance data. And in fact, we can decompose the entero signature concept into several parts. So we can look at the primary entero signature that dominates the environment, that is very similar to enterotyping. We can describe discretely the composition of entero signatures. So this sample has ES-Bact and ES-Firmicutes, for instance, or we can use the full quantitative information that we have. It's 30% ES-Bact and 70% ES-Firm. But what is really nice, I think, about the model, about NMF, is the reusability of the model. Let's say you want to add a sample to your data set, or you want to apply the model to another data set. In many cases, you would have to learn again. But with non-negative matrix factorization, you can reuse the W matrix that I showed you earlier, describing the entero signature composition, and apply it to a new abundance data. That's a non-negative least square optimization problem. And you will have the weight of the same entero signatures into your new data set. So here, what we show here on the new data set is that reusing the signatures that we tested. Of course, less than if we learn de novo on it, but still really well. Then we build models in order to have biological insights, right? So what we did here is to look at the association between the entero signature weights and clinical metadata that we had regarding our individuals, our samples. So we found several observations, but the most notable would be that the prevalence of ES-back to dominate treatments and the relative abundance of ES-firm is associated to very healthy outcomes, to healthy phenotypes. All right, so we built our model on an aggregation of several cohorts, 5,000 samples. But I must say that these cohorts, they mainly came from Western populations. And that is, of course, a problem because when you do learning, if you bias your learning the dataset, well, you will have bad predictions on other datasets. So the question was raised on whether the 5 entero signature model would be useful at all to characterize non-Western populations. So we gathered a few cohorts, 1,000 samples roughly, from rural populations, non-Western populations. So those are the orange cohorts here, the green ones being the original on which we did the learning. And we compared the fitting score of the model on those cohorts. And you can see that for most of the cohorts, it's really good. We have a very good fitting score. And then it's really bad for 3 of the cohorts. So we tried to understand what happens in these 3 cohorts. And we found a common feature, which is that the samples were ethanol preserved, which may have an impact in metagenomics on the microbial composition. But well, it happens in very rural areas. It's very complicated to store the sample in ice, right? So then why does the model works that well, because it's known that Frioux is dominated by Prevotella. And Prevotella is one of our entero signatures. So that's why it works that well. And if you look here at the adults microbiome composition in the non-Western and Western populations, you can clearly see the prevalence of the entero signature dominated by Prevotella. And that in Western populations, you have rather a codominance of Bacteroides and Firmicutes. Going back to these box plots here, you can see that we have long tails of outliers for which the model does not fit really well. So we call these samples ES atypical, and we try to understand why the model didn't fit. So we looked at the dominated genera in the samples. They were sometimes very pathogenic, and we tried to associate the fitting score of the model to clinical metadata that we have for the samples. And what we observe is that, for instance, you have bad fitting scores in preterm infants, so newborns that were preterms, newborns C-section delivered, or right after antibiotic treatments. So this model, it really aims at describing healthy microbiome, but by looking at how well it fits the microbial composition, you can identify perturbed ecosystems. So that can be useful when diagnosing dysbiosis, for instance. So if we summarize on this part, we built what we hope is a unified model for the gut microbiome composition. It is easily generalizable to other datasets. And in fact, we built a website on which you can upload your abundance matrix and directly get the entero signature composition of your datasets. We identified some interesting outcomes relating to clinical data association. But originally, I come from the field of metabolic modeling, so I couldn't help looking at microbial guilds. So I did a few analyses comparing how complementary to each other the guilds and entero signatures were. I won't have time to go into the details, but I shamelessly use it as a transition to the next part of my talk, which is going beyond the taxonomic composition and addressing the functional diversity of microbial ecosystem with metabolic modeling. So here I will again split my presentation in two parts. First, biology. So for those of you who are not familiar, we try to study the cell as a system of interacting subcomponents, the genome, the proteome, the transcriptome, etc. And that is quite complex by itself. But when you study microbial communities, it's even more complex because you have interactions between the microbial populations, and you have to take them into account because they will have an impact on the system itself. Fortunately for me, those interactions, most of them, they are mediated by metabolism, which makes metabolic modeling a very good start to get hypotheses on what happens in microbial ecosystems. So now is metabolic modeling 101 in a very small nutshell. So we start with the genomes that can be reference genomes or metagenomic assembled genomes. And we want to build metabolic networks out of that. So we rely on the structural and functional annotations of the proteins. And by using dedicated databases, we can associate functional annotations to metabolic reactions that occur in the cell. So a metabolic reaction would be something like that. A plus 2B gives 2C plus D. And at genome scale, so building genome scale metabolic networks, we have a few thousand of these reactions associated to a genome. So looking a bit closer to a metabolic network, here you would have in yellow the metabolites, and in blue the reactions that consume and produce the metabolites. So here it's already quite messy, but it's just 70 reactions. And we have a few thousand in a genome, right? So we need models to analyze them. So we build metabolic networks for each of the populations that occur in our microbial communities. And then we use them as input to feed models. So we have several kinds of mathematical models that use metabolic networks plus additional information. So for instance, environmental conditions, possibly a metabolic objective. And here you have at least two types of models that you can build. The most widely used is constraint-based analysis, flux balance analysis. I will say a few words about this later. And then that I really like, that is a qualitative model, a logic model of metabolism, and I will also present you what we do with that. So first, I will show you how we can craft numerical models for understanding the metabolism of microbial communities. And I will tell you a few more words about this numerical model. So what we try to do is to compute the metabolic activity, what that we call flux, in the metabolic network, so in the metabolic model, by assuming steady state in the cell. So no compound accumulates, the concentration of the compound is stable over time. And we include additionally thermodynamical constraints that enable us to have a cone of possible flux distributions here that we can study. If we make an optimization, that's flux balance analysis, maximizing, for instance, the growth, the biomass production of the organism, we can find optimal solutions and analyze the flux distributions within the system. We can build dynamic models out of this. So that's dynamic flux balance analysis by integrating at each time step the results of flux balance analysis into an ordinary differential equation. That way, we can follow over time the growth of several of the populations. So during the PhD of Maxime Lecomte in a project led by Hélène Falentin at STLO in Rennes, And with the help of Simon Labarthe and David Sherman, we got interested into what happens during cheese production. So in over a 7-week experimentation process, we followed the growth and metabolism of 3 bacteria here. And we had a lot of data to build models in order to understand what are the mechanisms responsible for the production of aroma compounds in the cheese. So we followed a 3-step process from the building of individual models to their calibration using pure culture data. And finally, we tried to predict the community dynamics. So the reconstruction of the metabolism of these strains in milk. And then we built the individual dynamic models that I mentioned earlier using pure culture experimental data. And you have here the prediction of one partner's growth and the pH in its growth culture. In orange, you have the experimental data, and in green, you have the simulation. It works well because it's the learning phase of the process. So we try to infer the parameters using the data. That overall, it works really well, except for a few metabolites at 3 time points. And this enables us to have the information on the concentration of metabolites over time, the metabolic activity, the metabolic fluxes that are associated to several molecules. So these molecules being the aroma compounds. that we could use offline in order to predict from input fluxes, so from the state of the environment, what would be the output fluxes for monitored functions. So doing that, we no longer have to solve optimization problems. Well, we did this for building the database, obviously. But then during the process of building the dynamic models, it's very, very fast. And this is what we did. So here you have the predictions of the dynamics of the infection. So concentrations of metabolites, here populations of the partners, transfers between compartments, the lumen and the cells. And in plain lights, you have the predictions you obtain with flux balance analysis, dynamic flux balance analysis. I forgot to mention that here at T equals 40 hours, that's when we simulate the infection of salmonella. So in plain lights, the ground truth, well, simulated ground truth. And in dotted lines, you have the results of the approximation with statistical learning. And you see that it is very close. Actually, we have less than 2% errors for a 60 times speed up. So that really is useful for us to build spatial model that we are currently in the process of finishing. So I won't show it now, but it really helps its computation. And now I will go back to this niche market that I mentioned earlier about an alternative model to study the metabolism. And it's actually very useful when you don't have the time, the energy, or the money to curate these models, to build very precise models, and you still want to get some insights into what could happen in the community. So what we do is the Boolean approximation of metabolic producibility. Remember the metabolic networks? So the circles here are the metabolites, the squares are the reactions. And here you have twice the same metabolic network. For each, we simulate what happens when we have a dedicated environmental composition. So here we have A and B in the environment, below C, E, and F. The network expansion algorithm that we use will compute the scope of these compounds that we call seeds, that is identifying the subnetwork in color here, that is activated, that is reachable from the environment. So from a single network, you can have several behavior depending on the environment that you model. So it's a Boolean approximation, so it's 0, 1, either it is reachable 1 or it's not 0. But it is actually quite convenient. And the way we model this is with answer set programming. So I know some of you know it, but well, this is even more of a niche market, I guess. So answer set programming is a logic paradigm from knowledge representation and reasoning. So we are no longer with machine learning, but we are with machine reasoning or symbolic artificial intelligence. And what is great about ASP is that you just have to describe your problem. You no longer care about how it will be solved. That really is the job of the solver. So given a problem, for instance, modeling the Boolean approximation of metabolic producibility, you will have a logic program that will be made of 2 parts. The first part is the one here. It is the knowledge database that you have, what you know about your system. My system is a metabolic network. It has reactions, each being characterized by the substrate, reactant, and its output products. And I have an environment here, the seeds. So that is the knowledge that I have. And then I describe my problem. My problem is to identify the sets of compounds that are reachable from the seeds. And that is just 2 lines. So that is very expressive, very convenient, and very efficiently solved. So that's why using these Boolean approximations and logic paradigm, we can solve a lot of problems very easily. And one of these problems is the work of Shabnam Ghesseminejad that was interested in the inverse scope problem. So what I showed you before is how to predict what is reachable in the metabolic network from the environment. The inverse problem is given a metabolic network and a metabolic behavior, a subnetwork that is activated, what would be the inputs? So the purpose here is, for instance, to help culturing some microbes that we struggle to culture in the lab. So ShamLab developed a tool, Seed2LP, that from a metabolic network will provide you with a set of seeds of metabolites that would be needed in order to ensure the producibility of targeted compounds. So the particularity here is that it is based on the reasoning approach that I described, but we are very well aware that people are not very familiar with this, and people like flux balance analysis. So what we did is a hybrid problem, ensuring that we combine what is great with ASP, so the scalability, the way we model the problem, and what is great with flux analysis, that is the precision. So the first question that we wondered is whether it is useful at all to use a model to answer this question, because there was a tool that was widely used that just looked at the structure of the graph of the metabolic network in order to answer the question of seed identification. So it used the structure of the graph, strongly connected components, and identified input vertices in the directed acyclic graph. And what we considered ground truth is whether the set of seeds that we obtained were ensuring flux balance analysis in steady state and flux in the biomass reaction in the 107 high-quality models that we retrieved from our database. Our goal was to find 1,000 solutions for each network in less than 45 minutes. So you can see here that for the structural tool, it doesn't work at all. Well, it works for 10% of the networks. As using a simple model that is not quantitative, but applying then afterwards a quantitative model, what it works pretty well. We are able to find seeds that satisfy steady state assumption. So here what we try to do is to set up all metabolites as targets, but in reality, we have a targeted behavior, a subset of metabolites that we want to ensure. So it's not exactly the same problem. And it turns out that it's a bit more complicated. So here you have the similar setup. So just using the Boolean abstraction and testing with flux balance analysis on the same networks for a targeted behavior. And you can see that we have 22% of networks for which we have no solution that satisfies steady state assumption, FBA. But for the rest, we have at least one solution that works and sometimes all the solutions that work. But I told you that we are working with hybrid models. So here we can directly guide the solving and keep only the models that satisfy steady state and provide the users only with those models. And this works for 91% of the networks. And for 8% of them, we just couldn't reach a solution in 45 minutes. So I will now go back to communities of metabolic networks. So I start with a very simple community here of 2 bacteria, a green one and a blue one that live in an environment that has 3 metabolites. What we want to do is to reconstruct metabolic networks for all genomes. So we have to do it automatically because the communities can be very large. We want to assess the complementarity between the metabolic networks. We want to understand what one metabolic network can provide the other. So here, there is one compound here that is producible by the green bacterium and is a precursor for the blue bacterium. That is the kind of motif that we want to identify. And in the case when we have targeted functions, for instance, here the production of G, we want to identify key species associated to the function. So for instance, to produce G, the blue bacterium is needed, obviously, but you also need the green bacterium because it provides this essential precursor, right? So we build a pipeline for this. That's called MetaG to Metabo. And it takes as input a set of genomes, reference genomes, or metagenome assembled genomes that represent your microbial community. We can provide automatic metabolic network reconstruction, or you can directly come with your own metabolic networks. And then the first step is to compute what is the individual metabolic potential for each of the populations. So what can they do in the environment that we simulate? Then we take into account the fact that these populations, they will interact with others, and some will produce molecules that will be useful for others. So then we expand the scope of reachable compounds, and that's the collective metabolic potential. If we compare both, we will find metabolites that are only producible through synergistic interactions, through metabolic complementarity. And finally, the last step of the pipeline is to provide community selection, starting from the initial and a set of compounds that I want to ensure the production of. What would be the minimal communities to reduce the complexity of the initial microbiome that would sustain this objective? So that's really a systematic screening of the metabolic potential. And I thought that maybe the best way for you to grasp what we do is to explain this on a very small, non-realistic example. So we have 6 very small metabolic networks, BACT1 to BACT6. And you have here the compounds, the reactions are the edges, so I simplified a bit. And the environment that we simulate is the yellow compounds. Here, the seeds are the yellow compounds, and we have green compounds that are the metabolic targets we want to produce with a simplified community. So the pipeline will give you the information of the individual scope of each population, what it can produce in the S1, S2 environment. And then you will have the added value of the interactions between species within the community. So here, the blue compounds, and you see that many more compounds are reachable when you take into account possible interactions. So I know I'm the last talk of the conference, so it's a bit too late for posters, but nonetheless, I know that they are available online as PDFs, and I really want to advertise the really good work of Juliette, who's here. She presented a flash presentation of her poster on the first day, and she worked on characterizing synergistic interactions within microbial communities composed of cyanobacteria, microcystis, and their heterotrophic partners during freshwater cyanobacterial blooms. So she used precisely this pipeline, and it enabled us to get some very interesting biological insights on the metabolism during blooms. So that said, our objective, as I told you, is to simplify the initial community to produce the green compounds. So the pipeline will give you the information that 3 bacterial populations, at least, are necessary to ensure the production of the green compounds. You can also have the information, the magenta arrows here, on what interactions would be needed for the community to work. And in fact, I told you that ASP is very efficient at solving logic problems, and you can enumerate all the solutions. So here you have 4 minimal size solutions. They are equivalent to produce the green compounds with 3 to 5 metabolic interactions. So if we look at the enumeration, we have these communities, and I used colors to distinguish the bacteria. So you might have noticed already that one of the partners occurs in every minimal communities. And we believe this is really interesting. So that's BACT1. We call it an essential symbiont. And it tells you that this population has some functional capabilities that are essential to produce the targeted metabolites. So that's really interesting. And then you have alternative symbionts that occur in some, but not all minimal communities. We were then interested into understanding how these symbionts, these key species, they interact within the minimal communities. So we like to build graphs, which we did. So we have an association graph connecting the symbionts if they occur together in a minimal community. And here it's nice and pretty, but in reality, the metabolism is so redundant and there is such a high combinatorics of solutions when you enumerate all possible minimal solutions that it's really dense and impossible to see. So we went a bit further in graph analysis and used graph compression techniques. So here, power graph analysis that enables us to compress specific motifs in a graph and build what I think is a good-looking graph, like this one, in which you can identify groups of nodes that actually have the same role in the communities. So here you see the essential symbiont here alone, and you see that you have 2 and 3 that are together in a node, and 5 and 6. And in fact, if you look at the combinatorics here, you see that you need backed one of the 2 between back 2 and 3, and one of the 2 between 5 and 6. So that really helps to grasp what happens in the minimal communities. So enough with the toys. We did this on real data. We worked with 1,500 metabolic networks associated to cultivable bacteria of the gut microbiome. And we had several sets of target metabolites that we wanted to ensure the production of. And we built these power graphs, and they were really interesting because they highlighted groups of equivalent species for the production of targeted metabolism. So here you will need one of the bacteria for from the yellow circle, one from the blue, etc. So that really helps the analysis. I think I'm soon running out of time. I'm close to the end. So I just want to show you what we are doing now. We are trying to go a bit further than just modeling synergistic interactions, and we want to be able to screen large communities and large number of large communities. So Maxime Lecomte again built a tool that aims at calculating synergistic and competition scores within microbial communities. So for the competition, we take into account the fact that several populations will consume the seeds or will consume exchanged metabolites. And the first test here on simulated communities that root leaf of soil microbiomes shows you that you have different behaviors between the microbial ecosystem. So the gut seems to be more competitive, the soil seems to be more cooperative. So that's still an ongoing work. So finally, to conclude on what we do to model the metabolism of microbial communities. So in the perfect scenario, we have small communities with curated metabolic networks, controlled communities, a lot of data, and then we can provide very precise numerical simulations, even go towards the path of digital twins, which is very trendy right now. So we do this with numerical model, possibly with machine learning, but we don't always have the possibility to do this. Sometimes we have large-scale communities, we have automatically reconstructed metabolic networks, and we are still interested into understanding what happens. So for this, we use machine reasoning, and we believe it helps working with the potential errors, the bias in automatically reconstructed metabolic networks, and still highlights some interesting features. So the two work together. We use the second to reduce the complexity and go towards curation and smaller community simulations. So I will finish by acknowledging the fact that everything that I presented is a teamwork. So I would like to thank all members of the Pléiades team. Some of them are here today, and especially those who worked on the projects that I presented, and all the other collaborators I worked with from Erlam and Quarium Institutes, from IRISA in Rennes, INRA in Rennes, and L'Abri in Bordeaux. And finally, I would like to thank the funders, the high performance computing platforms that are very useful for us, and you for your attention.